Starting with a new chapter, which is reproduction in plants. What is reproduction? Reproduction is the process by which one organism can produce other organisms of its type, where the parent can produce offsprings of its type. Reproduction can basically occur by two methods, which is asexual reproduction and sexual reproduction. Sexual reproduction is also known as amphimixis, wherein amphi or amphos refers to both, where two or both parents come together and mix to produce a new organisms, which shows characters similar to both the organisms. While in asexual reproduction, which is also known as apomixis, the reproduction occurs without mixing of the gametes or the parts of the parent. In angiosperm, vegetative propagation and sexual reproduction are the two ways in which the angiosperms reproduce. In some methods, in some lower organisms, however, different methods of reproduction are also seen, like binary fission, wherein a single parent divides into two organisms, which is seen in bacteria and amoeba. Other such method which is seen in lower organisms is by budding, wherein small buds are formed on the surface of the organism which break or get detached from the parent at a later stage and grow as a new organism, which is seen in organisms like the yeast. Another method is sporulation, which is by formation of spores, wherein spores are formed which may be motile or non-motile, which develop into new organisms at a later stage. In organisms like the algae, motile spores known as zoospores are formed, which are seen in organisms like Chlamydomonas. While in fungi, non-motile spores called conidia are formed, which is seen in organisms like the penicillium. The next method is by fragmentation, wherein a single filamentous organism breaks into several fragments which develop or grow into a new organism at a later stage, which may be seen in case of filamentous organisms like the Spirogyra. Now we'll be talking about some of the asexual reproduction methods or the vegetative reproduction or propagation methods which are seen in angiosperms, which you talked about earlier. First is by production of special organs called the tuberous roots. In some plants or in some angiosperms, the roots become swollen by storage of material or storage of reserve food material. These roots show presence of buds on their surface, which sprout, giving rise to leafy shoots and roots. The roots which may be formed may either be single or may be in clusters, depending upon which they may be called as simple or single or fasciculated roots, which are specialized structures which are developed for vegetative propagation in sweet potato like organisms or plants. Another such structure which is developed for vegetative propagation in plants is the stem tuber, which is seen in organisms like the potato or the Solanum tuberosum, when the terminal part of the root gets swollen due to storage of food material or reserve food material. The terminal part of these swollen roots shows presence of eyes or notches. These notches consist of a number of nodes on their surface and some axillary buds, leafy shoots or scale leaves. After termination of dormancy period under favorable conditions, one of the axillary buds gets detached or sprouts from the eye, suppressing the growth of the other buds. The tuber can be cut into pieces consisting of an axillary bud or an eye and are grown separately for commercial cultivation. So the use of a stem tuber consisting of an eye is that it can grow independently. Another type of organ which is developed for vegetative propagation is the runner, which is a slender, prostrate and subaerial branch which creeps horizontally on the surface of the soil, which is produced by organisms like the cynodon, dobe grass, Frageria or strawberry, and oxalis for vegetative propagation. Runner develops from the lower axillary bud of the stem and is thin, elongated, cylindrical, wire-like structure with long internodes. It creeps on the ground and becomes rooted at the end nodes, where shoots are produced from the upper side of the nodes which afterwards get detached from the surface and can grow into new organisms. Similar structures similar to runner, which are the stolon, offset and sucker, are also studied in angiosperms. Another type of structure which is developed for vegetative propagation is the leaf. In some organisms like the bryophyllum and kalanchoe, epiphyllous birds are observed, which are also known as foliac birds, which are produced at the notches of the lateral veins, and these birds start sprouting at the later stage on the leaf to form leafy shoot and adventitious roots. While such sprouts fall on the surface, they can grow into new organisms. In some species, specifically of bryophyllum, the new plants formed are from the leaves only when the leaf is separated from the parent plant and falls on the wet soil. So these are the four structures or organs which are specially developed in angiosperms for vegetative propagation. The first being tuberous roots, second stem tuber, third runner, and fourth leaf. Apart from the natural methods of vegetative propagation which we studied earlier, agriculturists and horticulturists have developed some artificial methods of vegetative propagation too, which can be broadly classified as CGB or BCG. The first being cutting, wherein a part of the plant or the organism, such as the root or the stem or the leaf, is cut or is separated and is grown into a new organism, which uses the property of an organism or an angiosperm of totipotency, which is wherein each part of the organism or the angiosperm can grow into a new organism. The second way of artificial propagation is by grafting, wherein a stalk or a plant is used or is grafted with a new organism, which is known as the graft or the scion, along with the cambium or the primary meristem, which can both then grow as a single organism. Thus, combination of characters of two organisms, that is the graft and the stalk, is possible in this method, which is highly essential and helps in evolution too. The third type of artificial method of vegetative propagation is budding, wherein instead of the entire organism, which is the stalk, which is the graft, a small bud with the underlying tissue is used and is grafted on the stalk. So it is somewhat similar to grafting, unlike instead of the entire organism, just a bud is used for grafting on the stalk. So these two methods, the cambium plays an important role because it is important that the cambium of the two organisms merges or mixes together and gives rise to a new organisms. However, this cambium is not found in monocots, and thus the methods of grafting and budding are not possible to be done in monocots. So these are the artificial methods of vegetative propagation. Talking about sexual reproduction in angiosperms, a flowering plant. For sexual reproduction specifically, a highly evolved and a modified shoot known as the flower, or the organ known as the flower, is produced. Flower though may have a different sense for us, but the actual function or the reason a flower is present in an organism is for sexual reproduction. The plant at the end where the flower bears basically consists of a pedicel with a swollen upper part, which is known as the thalamus. So this is the pedicel with a swollen upper part known as the thalamus. The thalamus shows presence of nodes, and the distance between the nodes is known as the internodes. So internodes and nodes are present in the thalamus. At the nodes, we see four different types of whorls, which are known as floral whorls developing, which are namely calyx, corolla, androecium and gynoecium. So the whorls can broadly be classified as essential, which are those which are essential for reproduction, and non-essential or accessory, which include the calyx and corolla, which perform function like attracting pollinating agents or protection, and the essential include androecium and gynoecium. The androecium consists of singular parts known as the stamen, gynoecium the carpel, calyx has parts or components known as sepals, while the corolla has components known as petals, which are colored and they are fragrant. Now the outermost part is the sepal or the calyx, inner to which is the petal or the corolla. inside which is the anther bearing part, which is the male wall known as the androecium or stamen, and the innermost is the gynoecium or the individual components known as carpel. Now we'll be talking about the structure of the anther or the stamen, which is the male wall or the male reproductive wall. The stamen basically consists of the filament, connective, and the anther on the upper surface. Anther basically is dithecous, consisting of two parts, which each part having two chambers, so it makes the anther tetralocular or having four chambers. Now we'll study the parts of the anther. The outermost part is the epidermis, which is the first part of the anther wall. It is the outermost common wall layer of anther, which consists of flattened cells. It is protective in function, so the function is protective. Inner to the epidermis, we have the second layer of the anther wall, which is the endothecium. It is common to all the four pollen sacs and consists of single layers of cell. The cells of endothecium shows characteristic fibrous thickness of callus. Cells of endothecium situated in the shallow groove between two sporangia remain thin-walled and represent the line of dehiscence. Fibrous thickenings of an hygroscopic nature of endothecium cells helps in the dehiscence. So the function is dehiscence at maturity. The next or the third part of the anther wall is the middle layer. It is internal to the endothecium and consists of one or two layers or three layers of parenchyma cells surrounding each pollen sac or the sporangium, which are the site for the production of the megaspores or the microspores or the pollen grains. They are called middle layers. The cells of these layers generate at dematurity, that is after the formation of microspores, and two pollen sacs of each lobe merge to form one anther chamber in each lobe. So they degenerate at a later stage. Then we have the tapetum, which is the innermost layer of the anther wall. It is the innermost wall surrounding the sporogenous tissue of microsporangium. Cells of tapetum are large in size and contain dense cytoplasm and one or more diploid nuclei or a polyploid nucleus. It provides nourishment or nutrition to the sporogenous tissue and developing microspores. It is also contributes in the formation of sporopollenin, which is a component of the enzyme. So it contributes in formation of sporopollenin and also in nutrition of the pollen sacs. The innermost part is the microsporangium or the pollen sac, which are sites for the production of microspores. In an immature anther inner to the tapetum, the microsporangium contains a compact mass or diploid sporogenous tissues. The cells of this tissue may undergo mitosis or they directly function as microspore mother cells. At maturity, microspore mother cells, which are diploid, which are found inside it, undergo meiosis to form four haploid microspores. Young microspores are generally present in the form of tetrahedral tetrads. Formation of microspores by meiosis or diploid microspore mother cells is called microsporogenesis. So the anther is the site of microsporogenesis. We'll be studying the structure of a pollen grain or a microspore which is produced inside the anther. A pollen grain is a unicellular, uninucleate, a spherical or oval haploid structure. It is with a double layer of wall called the sporoderm. So the outer layer is known as the exine, which is smooth or spiny depending upon whether the type of pollination is wind or insect pollination. And the intine consists of cellulose and pectin, which is found inner to the exine. The exine is mainly composing sporopollenin. The intine and the exine layer or the sporoderm shows presence of a small gap, which is known as the germ pore, from which the pollen tube is seen arising at a later stage. And generally, pollen grains may be uniporate in case of monocots and triporate, consisting of 3 germ pores in case of dicots. The inner layer of sporoderm is called the intine, as we talked about it earlier, and shows presence of the cellulose and pectin as its components. Now basic steps are followed in the angiospermic reproduction system or the sexual reproduction system in angiosperms. The first being microsporogenesis, which is formation of pollen grains or microspores inside the anther, which is the male wall. Then macrosporogenesis, which takes place inside the ovule or the carpel, which is the female wall. Then we have the male and female gametophytes formation, which is followed by pollination, and then fertilization, and finally formation of fruits and seeds. We studying the next step in the angiospermic sexual reproduction system or the process, which is the development of male gametophyte. The microspore or the pollen grain is the initial cell of the male gametophyte. The development of male gametophyte is endosporic, that is, it occurs within the microspore. It involves only two mitotic divisions, wherein the number of chromosomes or the diploidy or haploidy is maintained. It is completed basically in two steps. The first being before pollination, which occurs in the pollen sac, and the second being after pollination, which occurs on the stigma. Before pollination in the pollen sac, while inside the pollen sac, the protoplast or the pollen grain divides mitotically to form two unequal cells. The smaller cell is called as the generative cell, while the larger cell is known as the tube cell. The generative cell generally has a large nucleus, a thin cytoplasm, and shows presence of vacuoles, while the tube or the vegetative cell shows presence of a large vacuole, cytoplasm, nucleus, and reserve food. After pollination, we'll be talking about the second stage now. After pollination, the two-celled pollen grain is deposited on the stigma surface, which comes in contact with the sugary stigmatic secretions and absorbs it. Due to this, the volume of cytoplasm increases and creates a pressure which acts on the intine, which causes the intine to grow through the germ pore inside the ovary. So, from the germ pore, we see a pollen tube arising. Which is seen grow to the style of the carpel. Then we have the contents, that is first the tube nucleus or the vegetative nucleus going inside it, followed by the cytoplasm and the generative cell. So this is known as a partially developed or young male gametophyte, which is in the cell stage. The tube nucleus, the cytoplasm, and the generative cell all migrate into the pollen tube. The pollen tube grows downwards towards the ovule through the style due to chemical stimulus inside the ovary, which are stimulated by the synergids. The generative cell then divides by the second mitotic division, giving rise to two haploid non-motile male gametes. This pollen tube with two male gametes, thin cytoplasm, and a degenerating sterile vegetative nucleus represents the male gametophyte. Thus, it is highly reduced structure. This can be the diagrammatic representation of the after pollination process or the after pollination development of male gametophyte, wherein due to absorption of secretions, the intine grows inside the germ pore into the style in the form of a tube known as the pollen tube, which consists of the tube nucleus, the cytoplasm, and the generative cell, which later divides by mitotic divisions into two haploid cells, which are known as the male gametes. So a male gametophyte is basically a pollen tube which consists of a generative nucleus which is divided into two male gametes, a thin cytoplasm, and a degenerating sterile vegetative nucleus. So this is what a male gametophyte is. I will be studying the structure of a carpel, which is a single female reproductive whorl. It basically consists of an ovule at the base, which has a style which projects the stigma. The stigma is the site for the receival of the microspores or the pollen grains. After the stigma, the pollen tube is seen growing inside the style, and it then reaches ovule, which is the site for the production of megaspores, which are precursors to the female gametophyte. The ovule is generally anatropous, which is having a bent axis and a downwardly directed opening. And is known as the anatropous ovule, which is most common in angiosperms. Now we'll be talking about the structure of an ovule. this is the vertical section of an ovule. The ovule basically consists of two main parts, the stalk and the body. The stalk of an ovule is called the funicle or the funiculus, which is known to perform functions of nutrition, support and projection. Also conduction is what it helps in. So these are the four functions of the stalk or the funicle of a ovule. The funicle bears a major swollen part of ovule at its tip called the body of the ovule. The stalk or the funicle is attached to the basal part of the body of the ovule called the hilum. The body of an ovule consists of many parts, the first being the nucellus. It is the main central bulk of the ovule's body. The nucellus presents the megasporangium proper of an ovule. It consists of many diploid parenchyma cells. So this is the central bulk, which is the nucellus. Then we have the chalaza or the chalazial end. The basal part of the nucellus from where the integuments develop is known as the chalazial end. So this is the chalazial end or the chalaza. This end of ovule is called as the chalazial end. Then we have the integuments. So these two layers. These are the protective coverings of nucellus which develop from the chalazial part of the nucleus and surround it completely except a small portion at the opposite end, which is known as the micropyle or the micropylar end. There are two integuments, namely outer and inner, and therefore ovule is called bitegmic. So an angiospermic ovules are generally bitegmic, consisting of two integuments, which is the outer and the inner. Then we have the micropyle. The integuments leave a narrow opening at the terminal end of the nucellus. It is called as the micropyle. This end of nucellus is called as the micropylar end. So you have the micropyle. Then we have the embryo sac. In a mature ovule, this is the embryo sac. In a mature ovule, nucellus shows the presence of an oval-shaped haploid structure at micropylar end, which is called as the embryo sac or the female gametophyte. It consists of 7 cells and 8 nuclei. I repeat, 7 cells and 8 nuclei. There is a 3-celled egg apparatus at one end, wherein we have the egg in the center and synergids surrounding the egg, while on the opposite end we have presence of 3 antipodals or accessory cells, and in the center part we have presence of a secondary nuclei which is formed by the fusion of polar nuclei which fuse at a later stage before fertilization to form a diploid secondary nucleus. So generally embryo sac in angiosperms is monosporic, which is producing one type of spores, endosporic, producing spores inside, 7-celled and 8 nucleus, which is known as the polygonum type of an embryo sac. So we will now be talking shortly about the functions. The funicle is known to perform functions in nutrition, support, projection and conduction. The integuments are known to perform functions of protection. The micropyle forms the passage for the entry of pollen tube during fertilization, and also for the seed germination it allows the entry of water and radicle coming out of it. So for germination and fertilization we have the micropyle or the micropylar end. Then we have the egg apparatus, which is the female gamete, and after fertilization gives rise to diploid zygote which develops into an embryo. The synergids play an important role in fertilization and degenerate after fertilization. The filiform apparatus which is formed inside the synergids is known to attract the pollen tube during fertilization. The polar nuclei fuse with the secondary male gamete, giving rise to the primary endospermic nucleus, which is a nutritive tissue, and the antipodals are accessory cells which degenerate after fertilization. Two integuments present in an ovule are also seen in the seed in the form of a seed coat, the outer becoming testa, which is a hard coat, and the inner becoming tegmen. Testa and tegma. Talking about the process of the development of the female gametophyte, a diploid hypodermal cell at the micropylar end of the nucellus get differentiated to form the archaeosporium. Mostly this single-celled archaeosporium directly functions as the megaspore mother cells, which later gives rise to the megaspore. The diploid megaspore mother cells, which is gives rise to haploid megaspores by the process of meiosis. This process is known as megasporogenesis. Megaspores are generally arranged in a linear tetrad. Generally, the chalazial megaspore remains functional. So, a diploid MMC gives rise to 4 haploid megaspores or megaspore cells. The upper one of which is found near the micropylar end, remains functional. while the others degenerate. Which is found in the chalazion end, so remains functional while the others degenerate. Then later 3 mitotic divisions are seen, which give rise to the embryo or the female gametophyte. The first produces 2. Initially the cell consists of a nucleus and a cytoplasm, which undergoes the first mitotic division, giving rise to a 2-nucleate embryo sac. Then we have the second nuclear division, which gives rise to the 4-nucleate embryo sac. Later we have the third nuclear division. So here we have the first nuclear division, the second nuclear division giving rise to this, and then the third nuclear division. After the third nuclear division, we have 8 nucleus inside the cytoplasm, which get arranged in the form which is later seen in the embryo sac, wherein 3 nuclei move towards the chalazial end, 3 towards the micropylar end, and 2 remain in the center. The chalazial nuclei then assemble to form the antipodals, while the ones near the micropyle show presence of an egg nuclei at the center, surrounded by the synergids. The polar nuclei remain in the center, which before fertilization combine to give rise to the secondary nucleus. So this is what the embryo sac or the female gametophyte basically looks like, with the egg and the synergids forming the egg apparatus. We will be studying about pollination. Pollination basically is the process of transfer of pollen grains from the anther to the stigma, and it was first noticed or discovered by Camarius in 1694. Pollination can broadly be classified into autogamy and allogamy. Autogamy basically refers to self-pollination, when allogamy refers to cross-pollination. Autogamy can further be classified into autogamy again and geotonogamy. In autogamy, the transfer of pollen grain takes place from the anther to the stigma of the same flower, while in case of geotonogamy, pollen grains are transferred from the anther to the stigma of another flower lying on the same plant. So autogamy is generally seen in plants like pea, while geotonogamy is seen in plants like cucurbit. Another type of pollination is allogamy or cross pollination, which is further classified into xenogamy, wherein the transfer of pollen grains takes place from the anther to the stigma of another flower of another plant but of the same species. I repeat, another flower of another plant but of the same species. While in the second type of allogamy, which is hybridization, the anther to stigma transfer of pollen grains takes place from another flower lying on another plant belonging to a different species. Another flower, different plant, and different species. Xenogamy is seen in organisms like papaya, and plants like papaya, while hybridization is seen in cotton like varieties of plants. Talking about the agencies of pollination, where we talk about the agents which are responsible for pollination or aid in the transfer of pollen grains from the anther to the stigma. These agents can broadly be classified as abiotic agents, which is non-living agents, and biotic agents, which are living agents. Abiotic agents can be broadly classified into wind and water, wind being anemophily, or wind pollinated plants being known as anemophilous plants, while the second type is hydrophily or hydrophilous plants. Hydrophily is broadly classified further into 2 types, which are hyperhydrophily and epihydrophily. Hyperhydrophily is the type of hydrophily wherein transfer of pollen grains takes place below the surface of water, while in epihydrophily, the transfer takes place on the surface of water. Anemophily is the transfer of pollen grains through wind, wherein the pollen grains produced are large in number, are smooth, and are lightweight to facilitate transfer by wind. Anemophily is generally observed in plants like maize, grass, jowar, and sugarcane. While hydrophily is observed in plants like Zostera and Vallisneria. Biotic agents of pollination include the entomophily or entomon, which is through the agency of insects, ornithophily, which is through the agency of birds, and chiropterophily, which is through the agency of bat-like organisms. Entomophily is generally seen in plants like salvia, rose, ancestrum. Ornithophyly is observed in plants like Butea and Bombax. While Cheropterophyly, which is pollination through the agency of bats, is seen in plants like Cadum and Bahuenia. So these are the different agencies of pollination. So now we'll be studying the development of an embryo. The zygote basically, which is formed by fertilization, gets converted into a oospore by the formation of a wall. The oospore divides transversely to form a large basal cell. This is the oospore. So inside the oospore, we see the formation of a large basal cell on the micropylar end and a small apical cell or the embryonal cell on the interior of the embryo sac. This at a later stage develops to give rise to the suspensor above it, and the embryo is seen on the upper surface. The shape of the embryo is seen changing from being globular in the first stage to becoming heart shaped subsequently and horseshoe shaped at an even later stage. Several divisions takes place inside during this stage, which leads to the development of the embryo. Now, as we come towards the end of the chapter, there are a few important terms which we need to remember. Just parthenocopy. Mostly the fruits are formed by fertilization, but there are some species in which fruits develop without fertilization. This is called parthenocopy, and such fruits are called parthenocopic fruits. One example being banana. And these fruits generally don't have seeds and so are preferred by people. Two important terms again are apomixis, which is a form of asexual reproduction which mimics sexual reproduction, wherein seeds of the product, though being products of fertilization, these are seen in structures without fertilization. This is called apomixis, and such seeds are called apomictic seeds. While polyembryo is a case wherein several number of seeds are seen inside a fruit, like in case of lemon and several citric fruits, and this is known as polyembryony. Double fertilization in angiosperms. The process of formation of fusion, process of fusion of male gamete with a female gamete to form diploid zygotes is known as fertilization. Male gamete plus the female gamete come together, giving rise to the diploid zygote, which is primitive stage of the embryo, male gamete, which is haploid. The fusion of one male gamete present in the pollen tube with the egg and the other present in the pollen tube with the secondary nuclei of the embryo sac is called as double fertilization. So the significance is that one of the male gametes fuses with the egg of the embryo sac, while the second male gamete fuses with the secondary nuclei which is seen in the embryo sac. The first stage gives rise to a diploid zygote, which later develops into the embryo, while the second fusion or the second fertilization gives rise to the triploid primary endospermic nuclei, which further develops into the endosperm, which is a nourishing layer seen inside the seed. The first fertilization is known as syngamy, which leads to the formation of a secondary zygote or an oospore, while the second fertilization or triple fusion gives rise to a primary endospermic nucleus. After formation of the PN, which is the nourishing tissue or the nourishing layer, and the oospore, some changes takes place in the endosperm and the embryo, which are known as post-fertilization changes. Different types of endosperms are formed, and depending upon the mode of development, 3 different types of endosperms have been recognized, which are broadly classified as nuclear endosperm, cellular endosperm, and halobial endosperm. Now the basic difference between these 3 types of endosperms is that in case of nuclear endosperms, a large number of nuclei are seen formed inside the endosperm. However, the formation of cell wall doesn't take place simultaneously, but takes place at a later stage. So this is the nuclear type of endosperm. While in a cellular type of endosperm, the formation of nuclei, that is the division of nuclei, as well as the formation of cell walls takes place simultaneously throughout the endosperm. This is the cellular type of endosperm. While in the halobial type of endosperm, the first nuclear division is followed by the formation of a cell wall. However, the subsequent nuclear divisions merely show the formation of nuclei or the division of nuclei, but are not followed by cytokinesis or the formation of cell walls. So these are the 3 types of endosperms.